Hello dear viewers, welcome to e-learning. Subscribe to our SCOP Academic YouTube channel and turn on notification. We are on to the vaccines. In this particular video, we will go through the cost trends in vaccine development and manufacturing. Then we will see the vaccine registration and its approval process. Vaccine manufacturing has evolved dramatically over the last half century. New techniques for the manufacturing and the testing of the vaccines have transformed the manufacturing environment. New vaccines like multivalent conjugate vaccines are considerably more complex to manufacture than traditional inactivated whole cell ones. The increased sophistication of the manufacturing processes means that the cost of manufacturing has significantly increased in the last few decades. In addition, the regulatory environment has evolved to a point where as many as 500 quality control tests may be conducted in the manufacture of a single vaccine. Vaccine manufacture is highly capital intensive. A manufacturing facility alone will cost to 500 million billion at 7 September 2011. As manufacturing costs are largely fixed, large manufacturers may produce vaccine in massive amount. For example, hundreds of millions of doses every year to achieve economies of the scale in production. But scaling vaccine production requires a significant investment in time. Even for relatively simple processes such as vaccine packaging, up to 2 years may be required to install and validate new packaging machinery. Building a new manufacturing facility takes an average 5 years to complete and validate with regulatory authorities. The vaccine registration and its approval. Because of their biological nature and that they are largely administered to healthy individuals, the entire vaccine development and manufacturing process is regulated. Before vaccines are licensed, the three successive phases of clinical development must be approved by a national regulatory authority and may only proceed from one phase to the next upon approval of the national regulator. The regulatory processes of the vaccine under development, they will consist of the regulatory review and approval, that is the phase 1 clinical trial, the phase 2 that will involve the regulatory review and approval, the phase 3 that will the regulatory review and approval, the phase biological license approval application, that is the review on application of approval of the BLA, and the post marketing surveillance, that is the regulatory review of the adverse events. When a phase 3 trial has been completed, the manufacturer must apply for a license to sell the vaccine. The license application review is so thorough and complete that it takes between 1 to 2 years to complete. The regulator has the authority to refuse or withdraw a product license if the manufacturer is not compliant with the current regulations. Here you can see the regulatory testing of the licensed vaccines. It involves the purity testing, potency testing, sterility testing, stability testing, inspection of the manufacturing facility, and batch-to-batch consistency testing. After vaccines are licensed, manufacturing is strictly controlled by regulators who test and have authority over the release of each production batch of vaccine. The regulator tests the vaccines for safety, identity, purity, potency, and sterility. Regulators also monitor the consistency of the product from one production batch to the next. Inactivation and attenuation are also checked to ensure that the products does not expose to the risk. Regulators will subject the process and the product to multiple tests with redundant checks to ensure that the testing itself is yielding correct results. General safety testing is performed by injection of the final container product in the abdomen intraperitoneally of the mouse or the guinea pig. Identity testing is specific to the nature of the vaccine but can include neutralization of a live attenuated vaccine with an antiserum. The purity testing must demonstrate that the vaccine is free of extraneous materials including moisture and pyrogenic substances. The products used in the manufacturing of the vaccine must also meet the standards of the purity. The potency testing involves demonstrating that the vaccine confers protective immunity. The tests are specific to the vaccines being tested but often involve virulence challenges in an animal model or virus titration or other quantification of an antigen. It is also necessary to demonstrate that the potency of the individual component of a combination vaccine are preserved when combined because some of the antigens that can reduce the immune response to the others. Sterility is tested on both bulk and the finished vaccines. So for further readings, the viewers can go through the references. In this particular video, we had gone through the cost trend in vaccine development and manufacturing and the vaccine registration and its approval process. Dear viewers, thank you for watching. Take care and happy e-learning.